So I was instructed to put this slide in, so just so you know, I didn't come up with that idea myself, but I have no financial disclosures to give to you today, or arrangements that I have to disclose, rather. I also want to admit that I don't have any fancy graphics in my talk today, just to put you all at ease a little bit. It's going to be pretty straightforward stuff. I thought there would actually be something a little bit more official today, opening up this room, some champagne over the podium or something. But you notice I did have a little ribbon cutting, but I guess the dean didn't show up to do it officially. Trying to advance. There we go. So the other night at dinner, actually, with Tom and a couple of faculty candidates, we actually talked about maybe renting out Giant Stadium at some point for grand rounds. I thought that might, this is the new Giant Stadium, by the way, off the Jersey Turnpike, and thought maybe the 50-yard line would be a good spot, but that wasn't going to be very easy to arrange. And then Tom made one other suggestion that maybe somebody like Bruce Springsteen would be good to open up the new auditorium, and I thought, well, you know, that wasn't unreasonable, because if you put a little gray hair on him, maybe you might think that I might look like him. Nonetheless, today I want to tell you about two stories coming out of my laboratory, I think both with a recurring theme of genomic instability. And for those of you who haven't heard about Fanconi anemia, I think you'll hopefully appreciate by the end of our time together that it illustrates a great model for investigation of genomic instability, and it's sort of a coming full circle from the bedside. We all talk about bedside to bench and then back again. It's sort of the way I've always tried to think about things in terms of my scientific career and my clinical career. And then secondly, talking a little bit about the translational science of observations on an interesting protein encoded by the H2LV1 virus and its effect in p53 mutant cells. So we all know about this whole multi-step process going on to cancer, and of course the examples seen in various genetic models, including the APC colon cancer model, even that requiring multiple steps and needing the generation of genomic instability in order to get from the beginning of this little scheme, or the beginning from normal mucosa here in the colon, if I can use this properly, there you go, all the way to full-blown cancer. And so historically, cancer biology has really used these genetic models of cancer susceptibility as a means to understanding more general applications, more general lessons in cancer biology. And Fanconi really stands as one of these, at the top of the category of these familial syndromes of cancer susceptibility. I think most of you are probably more familiar with xeroderma pigmentosum, whose study led to the elaboration of the nucleotide excision repair pathway. And these are just some other examples as well. But Fanconi kind of stands apart within this special subset of diseases. First of all, I'll just point out that this subset of diseases is unique in the fact that they really represent hypersensitivity syndromes. That is, cells taken from the patients themselves and exposed to whatever specific or broad DNA damaging agent, these cells demonstrate a hypersensitivity profile. That is, they die at a far more rapid rate, or at doses that are lower than your typical doses that a wild-type cell will die at. Or similarly, you take the mutant cell corrected with the wild-type cDNA for which the cell is mutated for, and you can achieve a full correction of wild-type resistance to whatever agent you're looking at. And that's really the hallmark of all those diseases that I showed you in the previous slide. So a little bit of a background of Fanconi, just to bring everybody up to speed. There actually was a Dr. Fanconi. He was Italian-Swiss, and before I started working on this particular disease, I didn't know that that was actually the case. And I've got to close this little thing here. Okay. So this gentleman actually made a observation in the early part of the 20th century about a family that actually had bone marrow failure, congenital defects, and more importantly, went on to get cancer in the form of myeloid. These are patients that get myeloid cancer, and if they were lucky enough to grow up, get solid tumors. But it was really this original observation of a clustering of patients with bone marrow failure and birth defects that led to the term Fanconi anemia. And I'll just point out that even though these patients don't look abnormal at first glance, if you put them all in a room at a summer camp, for instance, where Fanconi families will often gather each year up in Maine, all these kids do end up looking like siblings in the same way that Down syndrome patients look alike. So it really is a syndromic looking facies. In addition, this is the classic thumb and radial ray abnormalities. I mean, these patients are, if they have any kind of thumb-like appendage at all, it's completely fleshy without any bone. As this X-ray bears out, there's no thumb at all, and in addition, there's actually no radius as well. So these are patients that are born with a whole array of different congenital defects, but most characteristically is this lack of a radius, lack of a thumb. On a biological level, however, these patients also have a problem handling DNA damage. This is really evident when these patients were first treated for cancers, and they literally had life-threatening or mortal injury as a result of chemotherapy. And when you examine their chromosomes on a cytogenetic level, you can notice that they really have a whole array of different abnormalities, chromosomes stuck on each other, radial forms, outright breaks, a whole panoply, but these can actually be quantified. And this is actually the diagnostic test for Fanconi anemia. So for all the pediatric hematologists in the room, if you order a test for, if you encounter a patient with bone marrow failure and you want to test them for Fanconi anemia, this is actually the test that you will get. But as I told you before, this actually is recapitulated at the cellular level, so much so that if I take fibroblasts from a patient or lymphoblasts or any cell type from the body and expose them to a DNA cross-linking agent, their cells will die at a more rapid rate. And again, this really is reminiscent of these familial cancer syndromes which demonstrate hypersensitivity to DNA damage. And it's that hypersensitivity to DNA damage that actually led to the elaboration of knowledge that there are actually 13 complementation groups. And in fact, each complementation group has yielded the cloning of a specific gene. But a lot of surprises came out of those original findings. First of all, 3 of those complementation groups are, in fact, well-described breast cancer genes. So all of a sudden here, we went from an obscure genetic disease called Fanconi anemia all the way to mainstream cancer biology, and the fact that breast cancer genes and their products are part and parcel of this pathway. BRCA2, BOC1, BRIP1, and PALB2. In addition, one of the other Fanconi proteins, FANCD2, actually binds to BRCA1. So there's really this incredibly interesting overlap of biology of Fanconi with the breast cancer pathway. And even though we know that BOC1, for instance, is a helicase, we know BRCA2 cooperates with RAD51 in the homologous recombination pathway, turns out most of the Fanconi proteins really don't have a definable enzymatic activity. So even though the prototype we've been following, xeroderma pigmentosum, from which we understood the nucleotide excision repair pathway, was sort of what led us down to this present state of knowledge, it wasn't going to be that simple. So we cannot actually take all of these gene products, dump them in a test tube, and come up with a repair-like activity. We're not advanced to that stage as of yet, although we well may be at some point. But this is what we do know about this pathway. In 2009, we know that 8 of the gene products in the Fanconi pathway reside in this core complex, which actually is dynamic, goes in and out of the nucleus in response to the cell cycle, binds to chromatin, but more importantly appears to modify the central protein FANCD2, which becomes ubiquitinated after DNA damage and during the cell cycle, and appears to functionally as well as physically interact with BRCA1, RAD51 in the homologous recombinatorial pathway. So that's kind of the present state of biochemistry. And that's kind of where my lab has been operating and trying to understand more about this biochemistry and really focusing on the central protein FANCD2, which again becomes ubiquitinated. And this ubiquitination is dependent on all these other proteins. In fact, it's been recently understood that this core complex has an E3-like ligase activity, which puts that single ubiquitin. So again, this is not a ubiquitination that results in degradation. Excuse me. Rather, it's a ubiquitylation that signals, and it signals this protein for downstream activity. So we've taken a strategy of purifying this protein, FANCD2, over a series of column steps, and we've been able to separate it out into 3 distinctive complexes over a gel filtration column: large, middle, and small, we term them. And really cool thing is that we can actually purify, going down one path, part of that little scheme I showed you, a single band of FANCD2 showing up on a gel. That's from endogenously purified FANCD2 on one arm. On the other arm, we can actually see the homogeneous tetramers of this protein by EM. On the other hand, going down a different arm of that chromatography pathway can purify a whole bunch of what we presume are going to be protein subcomplexes of FANCD2. And indeed, by mass spec, we can identify this band as FANCD2 and its binding partner, FANCI. And we have also identified a number of other proteins in this way, which I just want to give you a flavor for right now. In this immunoprecipitation Western blot, we actually demonstrated that FANCD2 binds to FANCJ, and I'll just remind you, FANCJ is one of those breast cancer genes, which are also Fanconi genes. FANCJ is BOC1B-RIP1, which is a helicase. And in fact, when you immunoprecipitate FANCD2 over here, you end up pulling down FANCJ in this Western blot over here. The interesting thing is that if you look at cells after the G1 phase of the cell cycle over here, when you get down into S phase, you lose that interaction completely. So you go from this interaction to no interaction over here. And we think that is because FANCJ is actually retaining FANCD2 in an inactive state. When you get to S phase and the replication fork encounters damage, then you have to free up FANCD2 to execute all of its actions, some of which I'll kind of come to in a few minutes. The other way the mass spec can really be a cool tool is, of course, to map post-translational modifications. We actually mapped an interesting phosphorylation site to serine 331 of FANCD2, and in fact, when we knock out that phosphorylation site, the resulting cells, which we transfect this construct back in, we can show that the serine to alanine mutant is completely non-functional. This is a survival assay where the wild-type FANCD2 corrects. The mutant cells are here, and of course, this phosphorylation mutant does not correct at all. We can also look by ubiquitylation of FANCD2, and I told you before that FANCD2 becomes ubiquitilated after DNA damage. This Western blot shows that cells that have wild-type FANCD2 go from non-ubiquitolated to ubiquitolated. You see that extra band showing up in response to DNA damage. Over here in the middle, there is a ubiquitylation mutant. That is that there's a K to R mutant, a lysine to arginine mutant, knocking out the ubiquitylation site. And you can see after DNA damage, there's no ubiquitylation. The phosphomutant, however, also demonstrates the same lack of ubiquitylation, indicating that it's been a functionally knocked out protein. The really cool thing is that FANCD2 also binds to BRCA2. And here, if you immunoprecipitate FANCD2, here's a wild type line. You pull down D2, you also pull down BRCA2. And it turns out that this phosphomutant serine to alanine completely knocks out that interaction as well. So this BRCA2 FANCD2 interaction, which I spoke of before, is actually dependent on this phosphorylation site. So we've actually gone on to map a number of different phosphorylation events in this pathway, summarized in this slide. Some of them are inducible by DNA damage. Some of them are inducible during mitosis. Some of them are inducible during both S phase and DNA damage as well. And so what we think is going on is that a summation of all of these different phosphorylation events are actually fine-tuning the DNA damage and or the replication fork movement during S phase. And so they all combine to fine-tune the DNA damage response. So we've actually mapped about 5 different sites in control of this pathway. We've also been able to identify interactions of the Fanconi pathway, not only with homologous recombinatorial machinery, but also with the mismatch repair pathway. 2 of the proteins on that particular silver stain gel I showed you before are MSH2 and MLH1, and in fact, when you immunoprecipitate FANCD2, seen over here, you indeed pull down MSH2 and MLH1. And in fact, this particular interaction seems to be dependent on DNA, to occur or be increased in the presence of DNA damaging conditions in this plus lane right over here. And in fact, if you knock out D2, you see that there's an overlapping increased sensitivity to cross-linkers, mitomycin C, over here. In this particular case, we're doing this with siRNA technology. We can knock down D2 and create sensitive cells. We can knock down MSH2 and create sensitive cells. And a really cool, interesting finding we've come up with recently is the fact if you knock down both of these proteins, both of these genes, the resulting, if you see here in lane 2, both of them are actually diminished in expression. You create a resistant phenotype. This is actually going to be a new source of investigation for my lab in collaboration with Sherm Weissman in genetics. I'll tell you a little bit more about that at the end of the talk. We've also shown that RAD18 binds to FANCD2, and this is yet another protein we found in our mass spec screen, or at least its binding partner RAD6, where we can immunoprecipitate FANCD2 and we pull down RAD18. And in fact, when we knock both of those down together, we can show that they're overlapping in their sensitivity, meaning that they're probably epistatic in their biological relationship. In fact, if you knock out RAD18, you can see here that you pretty much all but eliminate the ubiquitination of D2 that you can see over here in these wild-type cells. And the other really cool thing is that you prevent the FANCD2 protein and its partner, FANCI, from going to chromatin at all until DNA damaging conditions have occurred. And this chromatin localization turns out to be as important as FANCD2 ubiquitination to the normal biological function of this pathway. So if you kind of look at the overall view of this Fanconi pathway, there's actually a dynamic localization of the various complexes involved, and showing you data. This is actually a summary of data from the lab over the last few years, showing that this coordinately goes in and out of the nucleus in response to phosphorylation, and that this complex interacts functionally with this D2 protein, which also binds to I and J, as I showed you before, and binds to its other partners, BRCA2 and BRCA1, controlled in a single transduction manner through ATR and CHEK1. Functionally, though, what we think is that the DNA damage response is actually activating the ATR kinase CHEK1 axis of the DNA damage response, which then phosphorylates FANCD2. ATR and CHEK1 both are thought to phosphorylate FANCD2 and FANCI, which then triggers a whole bunch of downstream events. Now, just to remind you, these proteins don't seem to have any functional domains. What we think rather is that they're actually interacting with a whole host of different S-phase specific events, including homologous recombinatorial repair, mismatch repair in what we would hypothesize to be an activating manner, and with replication machinery in the form of the MCM27 helicase and transcriptional machinery, both of which we believe will be an inhibitory manner. So in the last couple of minutes, just want to tell you about a little bit of, I guess, a paradigm for why you should go with colleagues to have a cup of coffee. And a colleague of mine, John Sems, who's now at Eastern Virginia Medical School, is a virologist who was working with me before in my previous life at the University of Virginia. And I didn't think I would have much in common with a virologist. But he was working on this HTLV1 tax protein, which turns out to be linked to adult T-cell leukemia, tax1 being a viral oncogene. And tax1 being an interesting protein which promiscuously activates a whole range of different kinases. And then he showed me some data that he generated in his lab showing that when you throw this protein into a cell, you actually promote genomic instability. It's actually a visual thing. You can see these little chunks of micronuclei coming off. And even more interestingly, he showed me that if you just threw this into p53 minus cells, you could actually increase the sensitivity of these cells. And this is just these are cells that are dying. I know it's a different way of looking at survival. And in each of these different doses of UV, you could actually increase the amount of cell death induced by these different doses. So this, the oncologist in me kind of clicked in thinking, well, is this a way we can manipulate a p53 mutant background P53, of course, being an important means of resistance in clinical oncology. So we did just a couple of simple experiments. That was actually taking some P53 competent lines, MCF7, and just using shRNA to knock down P53. And in fact, in these P53 knockdown cells, these cells are actually resistant. But if you throw in this protein Tax1 by gene transduction, in this case, you actually make the cells more sensitive. Contrastingly, here is the original MCF7 line, which is sensitive. You throw Tax in, they become resistant. And this is actually a therapeutic dream. In the case of a tumor, which is P53 mutant and resistant, you can create sensitivity, but sparing presumably P53 wild-type patients and their cells, sparing them the toxicity from the therapy that they're undergoing. It turns out that Tax promotes a pro-apoptotic array of increased gene expression, and all of these apoptotic proteins are increased in expression in the presence or minus of a DNA-damaging agent, PUMA, NOXA, and then the dimerization of BAX down here. They're all increased in expression. So what we believe is going on, as I mentioned to you before, that Tax actually promiscuously interacts with a variety of different kinases. In this case, we have actually data showing that Tax interacts with CDK4, which we believe is counteracted by the effect of P53 increased expression in the context of DNA damage, which is actually promoted by another Tax interaction with CHEK2 over here. However, when you don't have P53 around, you really don't have that arm of the pathway anymore. And of course, mutant P53, an important clinical issue. But what happens with Tax and CDK4 is you probably drive the cells through into S phase because of the phosphorylation of RB. You free up E2F1, and you also upregulate a pro-apoptotic program. And the cells go into S phase, and then they're susceptible to DNA damaging agents, specifically S phase agents. So what we're trying to do here is to try to adapt this biological activity into a means of eventually trying to make chemotherapy approaches, just standard chemotherapy approaches, more effective. What we're in the process of doing is seeing what different types of P53 backgrounds that this effect will be seen. It turns out to be not all, but a significant number of them. We're also trying to actually hone in on the one domain of Tax that will actually serve as a minimal domain for this effect, and think about using the potential for Tax peptide, but also doing a small molecule screen to try to see if we can actually exogenously, with the use of a small molecule, activate CDK4 to simulate that Tax effect, and then eventually get down to treating tumors in new mice before trying to get back to the bedside and the patient. So I just want to thank the people in the lab who've been doing the work. Andrei Tomaszewski, Natalie Collins, Gang Z, Valia Mihlova, who's done the Tax work, Stacy Williams and Allison Green, Stacy who's done the RAD18 work and the mismatch repair work, Allison who's done the transcription work in Fanconi anemia, and Xiaoyang Chen, who's done work in actually all the different projects that I've described for you today, and collaborators in Liverpool and Eastern Virginia. And then I guess I know the point of having Cancer Center Grand Rounds is to really promote the fact of the potential for interactions. But in just a short 2.5 years since coming here, a lot of those interactions for me have already been happening. It's really been the most exciting part about coming here, among the most exciting parts about coming here. But these are active collaborations going on right now for my laboratory. I mentioned Sherm Weissman has been helping us in a joint effort to set up a Fanconi shRNA suppressor screen. That is to identify genes which, when you knock them down, will suppress the Fanconi sensitivity phenotype. Peter Glazer, been working with on mismatch repair. Patrick Sung on FA biochemistry. Diane Krause on another bone marrow failure syndrome. And Rick Lifton on another project we have going on looking at the genetic basis of Hodgkin lymphoma. So with that, I want to thank you all very much.